We are going to go ahead and get started. How do I? Perfect. Okay. Hello, everybody. I am Inka. For those of you that don't know me, I'm one of the mods here. I'm also a PhD student in neuroscience. I'm going to be giving the presentation on cellular mechanisms of plasticity today. We are going to cover a lot of things. It's about 20 minutes. I timed it. A couple things to note before we get started. I'm going to draw on a couple of references throughout the talk. The citations that I'm using are going to be in blue, either in the top right corner or sometimes it's like in the bottom corner. Just to, if you want to go back and reference it, the slides were posted a little bit higher up in the chat earlier before we started. Another thing, the active figure, the figure that I'm talking about, is going to have a blue box around it. I know there's going to be a lot of figures, so that's just kind of to help you follow along with what I'm saying. Feel free to stop me at any time to ask questions or to comment. This is not a lecture, and I am not your professor. Okay, we're going to go ahead and get started. Okay, so we all come from different fields that interact with the concept of learning in many, many different ways. So I always like to start out this kind of presentation by leveling the playing field and asking what everyone thinks is a good definition for learning. Would anybody like to volunteer one? We have a chat if you want to type in it. Memorizing a pattern. Yeah, that's a good one. See a lot of people typing. Gaining new information, retaining data, adapting to one's environment, building a habit, forming associations, strengthening neural pathways. Yeah, those are all really great. Well, to me, learning is the strengthening or weakening of associations. To my cat here, where, what? Oh, I have to tap it. I don't know how to do this. To hello, to my cat here on the right, she has learned to associate this window with hummingbirds. So every time she sits in her favorite spot, that memory gets strengthened a little bit. On a cellular level, this could be the changing of the ratio of the strength of two synapses. You see this one, they both start out equal, but over time, one of them gets stronger and the other gets weaker. Where's my laser pointer? Yes, there we go. One of them gets stronger, the other gets weaker. Or it could be strengthening one connection and letting all the others die off. We see this most often in neurodevelopment. Sometimes it's changes in white matter volume between brain areas. We see this in things like epilepsy and sometimes in Alzheimer's. In some cases, we strengthen entirely different connections. In fear memory extinction, we actually strengthen the inhibitory control from the prefrontal cortex instead of weakening the fear memory from the thalamus to the amygdala. Okay, we're going to dive a little bit deeper into the cellular mechanisms. That's what you're here for, after all. The most important thing to understand whenever we're talking about synaptic plasticity is that it is a reversible process. That means that the same processes are going to happen forwards and backwards to change the strength of the synapse. Most often, that process is going to be the addition or subtraction of a phosphate group to change the nature of a protein. But in general, the end result is going to be a change in the number of AMPA receptors available for signaling. There are two major forms of plasticity. Those are going to be potentiation or upscaling and depression or downscaling. Potentiation is going to be the form that strengthens the synapse. It usually happens when there's a high influx of calcium due to the activation of NMDA receptors, and it's going to lead to us having more AMPA receptors in the synapse. Depression, on the other hand, is going to weaken the synapse. The cause of depression is a little bit contentious. Some data says it's due to low to moderate calcium influx, but other data says that it's going to happen even when ions aren't allowed to flow through NMDA receptors. In general, though, depression is going to decrease the number of AMPA receptors in the synapse available for signaling. You may have also heard of long-term potentiation and long-term depression. These are similar to the short-term forms, but the long-term forms involve changes in gene expression. One of these changes could be making more AMPA receptors for insertion into the synapse. In general, though, the long-term forms are going to have longer lasting effects, and they're going to be slower to activate. Okay, spike timing dependent synaptic plasticity, try saying that 5 times fast, is probably the most important form of plasticity that we're going to talk about today. But to understand it, we really need to understand NMDA receptors first. NMDA receptors are glutamate receptors that let cations into the cell. What's different about them is that at rest, a magnesium ion is going to sit inside the channel pore and block cations from entering the cell. It sits there because it's attracted to the negative interior of the cell at rest. When the cell is depolarized to around negative 50 millivolts by intracellular cations, the magnesium is going to be pushed out of the cell. This is going to allow the cations outside the cell to enter the cell and trigger an action potential, as well as the short-term forms of synaptic scaling. NMDA receptors are considered coincidence detectors because they only activate when there is activity in both the pre- and postsynaptic cells, and it's this function that makes them really important for plasticity. Okay, we are going to get into our first discussion question. I thought I'd add this to nope, not potassium, it's the magnesium. I thought that I would add this just to kind of boost the engagement. So, based on how NMDA receptors work, how do you think spike timing dependent synaptic plasticity strengthens weakly active synapses and weakens inactive synapses? Remember, weakly active synapses are those that wouldn't be strong enough to push out the magnesium block. Give y'all a couple minutes to talk about that. Is it because NMDA receptors allow for the influx of calcium and that makes the voltage go higher? Not quite. You're close though. Okay, that was a good guess. So what's actually happening, where's my clicker? What's actually happening is as an action potential is going to travel from the active synapse to the cell body, it's going to take a little pit stop in another dendritic spine and push out the magnesium block in spines that aren't strong enough to push out the magnesium block by themselves. This is going to lead to strengthening of the active synapses, even weakly active synapses, and weakening inactive synapses. It is also going to provide a cellular mechanism for classical conditioning, right? The bell wouldn't normally be strong enough to cause an action potential, but when it's paired with the food, it is, and that synapse gets strengthened. Okay, that's great and all, but glutamate isn't the only neurotransmitter that our cells use, right? Neuromodulators are a special kind of neurotransmitter that don't open ion channels or directly affect the charge inside the cell. These are important for everything from sleep-wake cycles to attention, like the attention you're probably paying to the Subway Surfers gif in the middle, and most importantly for us, to memory. There are lots of different kinds, but we're going to focus on PKA for today. Dopamine and norepinephrine are the primary neuromodulators that activate PKA. PKA is a protein pathway, and it's stimulated by dopamine binding to either D1 or D5 receptors, or norepinephrine binding to beta adrenergic receptors. This is going to activate a protein called adenylyl cyclase, which is going to produce cyclic AMP. This cyclic AMP is going to activate protein kinase A by separating its regulatory subunits from its catalytic subunits. The catalytic subunits are then going to cause the cell to release more neurotransmitter by blocking potassium channels or opening calcium channels. In the long-term form, PKA is going to activate CREB, which is a protein that changes gene expression. If you remember before, this is the primary mechanism for long-term synaptic plasticity. CREB is going to produce a protein called ubiquitin hydrolase, which breaks down the regulatory subunits that block PKA. This is going to produce what we call persistently active PKA that's going to increase the neurotransmitter release of a cell for a long time. CREB is also going to transcribe genes that allow for the growth of new synapses. PKA is also going to have some postsynaptic effects that we're going to talk about next. Okay, so one of the primary mechanisms how dopamine promotes synapse strengthening is through the activation of PKA. You can see when dopamine is applied in the red, we're going to see synaptic depression, which is this decrease in activity, instead turn into synaptic potentiation. CREB is the cyclic AMP response element binding protein, which is a mouthful. Hydrolyse is inside lysosomes. Hydrolase is an enzyme. Neuromodulators and neurotransmitters are the same. There's a couple of different categories of neurotransmitters. Neuromodulators is just the specific word for those that don't like open ion channels or change the charge inside the cell. Okay, when we see dopamine applied to a cell, we're going to see synaptic potentiation, strengthening the synapse instead of the weakening. Yeah? We see the opposite effect when we block dopamine. When we apply dopamine antagonists, that's in the black, we're going to see synapse weakening instead of synapse strengthening. Controlling glutamate reveals what's actually happening. When we block NMDA receptors, it stops dopamine from turning synapse weakening into strengthening. So here is the normal weakening. Here is dopamine without the NMDA blocker. And here's dopamine with the NMDA blocker. Okay? So we don't see the synapse weakening that we normally have, but we also don't see the strengthening that we have with glutamate. If we add forskolin, which is a PKA agonist, it's going to increase PKA activity the same way that dopamine would. We see the same exact effects as dopamine, which really proves that dopamine is working through PKA. Now, this data is going to show two things. First, there is a two-part system for synapse strengthening. Dopamine is going to reverse the effects of synapse weakening, and NMDA receptors are going to cause the synapse strengthening. The second is that dopamine reverses synapse weakening through activating PKA, and that this mechanism can be generalized to any neurotransmitter coupled to the pathway. Sometimes we see acetylcholine or serotonin, norepinephrine, dopamine. Sometimes we even see large proteins like orexin that are going to activate PKA. And so anytime you see PKA, you can see this effect of learning. It's a little bit different, but neural strengthening it's part of learning. It's a necessary process for learning. Is it possible to have too much or too little of one? Yes, it is. Okay, bye, doctor. Okay, now we're going to get into the actual nitty-gritty molecular details. So this is a diagram of everything that's going on in the postsynaptic cell. It is a mess of proteins if you don't know what you're looking at, so we're going to break it down into its most important elements. So PSD and SAP are proteins that make up the postsynaptic density. The postsynaptic density is a dense web of cytoskeletal components that anchor signaling proteins to the synapse. It's just kind of this, you can see it here on this electron micrograph. It looks really dark and dense. This is the postsynaptic density. It's just where all of the AMPA receptors and all of the proteins that keep them there are kept like really close to the end of the synapse. Receptor anchoring is a really complex topic. I could honestly spend an entire Neurotalk on just this topic alone, but for your sake and for mine, we're going to move on. Can the density vary? It does vary by synapse type and synapse strength, but it doesn't necessarily vary from person to person at baseline. So PSD is one of the proteins that anchors AMPA receptors, and it's going to have a specific protein modification called palmitoylation, and this modification is going to let it hang onto the cell membrane. By hanging onto the cell membrane, PSD is going to stabilize the synapse by preventing receptors from moving around just all willy-nilly. Glutamate receptors are going to be attached to these PSDs through interacting with the PDZ domains. That's not going to be on the quiz later. Some receptors, like NMDA receptors, can attach directly to PSD, but some, like AMPA receptors, need a special connector protein, like TARPs. There's no quiz, I was just joking. No, so tau is going to be a connector protein that's found in the axon microtubules. It doesn't really play a role in the terminal buttons or the dendrites because there's mostly actin there. Tau interacts with like tubulin. No, sorry. I love answering questions. Okay, so PSD anchors the synapse and stabilizes it by not letting things move around, right? Another function of PSD is to align receptors with neurotransmitter release sites. This alignment is going to decrease the amount of neurotransmitter that the presynaptic cell has to release and lowers the risk of cells dying from being too excited. Normally, vesicles are going to be tethered to calcium channels during neurotransmitter release through a connector protein called RIM, R-I-M. From being too excited. So when a cell is having too many action potentials, we get what we call excitotoxicity, which is where the cell is going to die if it doesn't get to rest. And so by reducing the amount of neurotransmitters the cell has to release, we reduce the risk of the cell being too excited because it's more sensitive to the neurotransmitter. Not quite being in survival mode. It's like if you were like running like constantly, there would be a point that you would be like, you would probably pass out because you were running so much, right? The cells are the same way. Yeah. It's actually one of the primary mechanisms for stroke is that whenever cells are dying during stroke, they're going to release all of their glutamate and kill the nearby cells that aren't necessarily deprived of oxygen just because there's way too much glutamate in the environment. So there is a mechanism against it. Most of the time, it happens because of um, like trauma, like uh, hypoxic events when you don't have enough oxygen, or if there's some kind of necrosis. Inflammation can also cause hyperexcitation. It's a very short period, and that's because there's such a large increase of calcium, and calcium is going to tell the cell to apoptosis or kill itself. Not really in cancer cells because most of the time they turn off their apoptosis mechanisms. What can happen is if they're really active, first of all, there aren't any neuromas. That just doesn't exist because neural cells don't divide. It's always gliomas, but most of the time cancer cells are going to turn off their apoptosis mechanisms. What could happen is if they're too active, they would have the accumulation of free radicals, which would damage the DNA. And if it gets damaged enough, the cell just can't divide anymore, so the cancer cell would die off. Danksies. Okay. Okay. So vesicles are tethered to the cell membrane and calcium channels by a protein called RIM. And that's going to bring them really close to this synaptic machinery that's going to be responsible for the neurotransmitter release. Calcium is going to flow in through this channel and trigger the release of neurotransmitters at the end of an action potential by activating these snare proteins. If you've never heard of the snare complex, I highly recommend you look into it. It's really a foundational concept in cellular neuroscience. It's one of the first things that I ever learned, and it's really important for understanding kind of why the cells signal the way they do. Okay, it turns out that RIM, this protein that kind of tethers to the vesicles, and PSD, which anchors the receptors, are found really close to each other, right across from each other in the synaptic cleft. It's unclear. Hi, Vincent. You're a little late, but better late than never. It's really unclear what connects RIM and PSD across the synapse, but it might have to do with the extracellular parts of AMPA receptors. We're going to call these pockets of neurotransmitter release and reception machinery nanocolumns. So, like, you can see there's large overlap here and here and here. Each of these is going to be a nanocolumn where we find both the vesicles and the receptors. But that only explains how glutamate receptors are anchored in the synapse. How do they actually get there in synaptic scaling? Receptors can get to the synapse in one of two ways. First, they can start in the extrasynaptic space, where receptors that aren't involved in signaling are kept, and then move through the perisynaptic space and into the postsynaptic space, where they anchor to the PSDs. The second way is for receptors found in recycling endosomes, right here. These are normally receptors that are just synthesized. They're newly synthesized. The cell just sent the RNA to the dendrites, and it just found its ribosome partner and got inserted into this endosome. A neuromodulator, like norepinephrine in this case, is going to enter into the cell and then into the recycling endosome, where it activates PKA. PKA is going to cause the insertion of the AMPA receptor from this recycling endosome into the synapse through that same kind of pathway that we just talked about for the extrasynaptic receptors. PKA is going to play an additional role in this process, though. Glutamate receptors are normally attached by this tail to the membrane, which doesn't allow them to move around. By phosphorylating AMPA receptors and NMDA receptors, PKA is going to release the tail and let them move around the cell membrane. This makes it possible for them to move to the synapse during potentiation and increases how much they bind to PSD, which is going to reverse the effects of synaptic depression. PSD is also important for this mechanism we just covered because it brings PKA close to glutamate receptors. It attaches to a specific protein called A kinase adapter protein, which you also don't need to know. But this is just going to bring all of the machinery for synaptic potentiation with PKA and synaptic depression with protein phosphatases close to the receptors. So it can happen really quickly, and it can do it with a lot of spatial specificity. Okay, we're going to get on to our next discussion question. So keep in mind that synapse weakening and strengthening are the same process, just happening in opposite directions. How do you think the postsynaptic density changes to weaken the synapse? You can type in chat or just talk. Something to do with calcium? Actually, yes. Maybe get rid of PSD. Yeah, those are great guesses. So it's actually going to be both. If you remember, receptor anchoring and localization through PSD is an important mechanism of synapse strengthening, so it should be no surprise that removing PSD from the synapse is important for synapse weakening. In synaptic depression, that palmitoyl group that tethers PSD to the membrane is going to be removed, which causes the PSD to come loose and float around the synapse. After that, calmodulin bound to calcium is going to wrap around the free end of PSD, that tail part, and prevent it from reattaching to the membrane. And after we remove some of the PSD, there are less spots for glutamate receptors, which makes the synapse less sensitive to that glutamate release. Okay, we're going to move on to some of the disorders affecting plasticity. Before we do that, does anybody have any questions about the cellular mechanisms that we just talked about? No. Alrighty. So there are so many disorders that are characterized by dysfunctional plasticity. We could spend all day just talking about these figures alone. But just for this talk, I'm going to focus on Fragile X and Alzheimer's. If you want to see it in the future, I could do another talk on some of these. I know quite a bit about schizophrenia and ASD, a little bit about epilepsy, but we'll leave that for another time. So, Fragile X syndrome, or FXS, is a neurodevelopmental disruption which is caused by a particular gene called FMRP, or the Fragile X mental retardation protein, being silenced. FMRP is going to primarily stabilize mature synapses by blocking certain proteins. You can see that here. FMRP inhibits synaptic protein translation, which normally leads to internalization and weakening of the synapse. Okay? So, in normal healthy cells, the extracellular matrix, the kind of cytoskeleton that wraps around the outside of your cells, is going to stop the growth of new synapses and keep the cells mature. In FXS, though, a protein called matrix metalloproteinase that is mostly used in neurodevelopment is going to be produced it's just going to go wild. And MMP is going to break down this extracellular matrix. So, based on that, what we just talked about, what do you think would be the result of breaking down the ECM and just letting MMP go wild? Not quite like a Thanos snap. It's more like a cage, and then the MMP is going to eat away at that cage and leave little holes for things to get out. The synapses can't strengthen, not quite. That's a good guess. Unchecked, like there's no control over it. It's just having a field day. It's going wild. Those are really good guesses. So the ultimate result of unchecked MMP activity is that the neurons of people with FXS don't ever mature. This is going to result in the primary symptoms of Fragile X syndrome. You can see here the kind of gray color is going to be FXS, and the black color is going to be the just normal healthy controls. You can see As the animals move from young to adult, we're actually going to see a change in those black bars between the two conditions. Yeah? But in the gray bars, we don't see that change because the cells never develop mature synapses. Okay, we're going to move on to Alzheimer's disease. Does anybody have questions about FXS before we move on? No? Okay. So in Alzheimer's, unlike FXS, we don't see too much plasticity, but too little. Before we get into this section, I have to say that a lot of Alzheimer's research is just too hard to follow if you aren't in the field because of all of the jargon. So to make it a little bit easier to understand, and thanks to Vincent for this idea, I'm going to be replacing the jargon with easy to understand placeholders. The first of those placeholders is going to be this evil cat. Evil cat is going to be representing the beta amyloid oligomers, or the bad amyloid for today. Anytime you see his face, it means bad amyloid. So good amyloid, the A beta monomers, is actually really important for your cells. Animals that have it removed from their genomes are going to die before they're even born. The main difference between this good amyloid and bad amyloid, besides for their structures, is that while good amyloid increases cell survival and plasticity through things like activating CREB, bad amyloid is going to block this pathway and kill the cells. One of the ways that good amyloid promotes plasticity is through causing CREB to be phosphorylated. If you remember from earlier, CREB is phosphorylated by pKa, and it's really important for plasticity. When bad amyloid is added to cells, right here you can see this bad amyloid over here, there's much less CREB phosphorylation than when the good amyloid or the pKa agonists are added. And this is going to result in decreased things like BDNF and other genes from being produced. And BDNF is really important because it keeps your cells alive and also kind of increases the neurotransmitter that's released. Okay, here's the second of our placeholders. This angel cat is going to represent good amyloid. So anytime you see angel cat, that means good amyloid. So looking at the graphs on the right, you can see that the bad amyloid right here, when compared to the good amyloid, results in decreased signal strength, which you can see on this graph, right? This is much lower than this one, significantly lower. And this really shows that the bad amyloid is causing synapse weakening. So based on what we talked about earlier, how do you think bad amyloid causes synapse weakening? And hint, it's not by blocking CREB. Think back to the primary mechanism we talked about for synaptic depression at the very beginning of this presentation. Inhibits pKa, maybe. Clumping of bad amyloid. I think we have a recording. We can probably send it in the chat later, but I think Wizzy's recording today. I'm giving presentation. Something with magnesium, maybe. Okay, now I have to introduce our last placeholder. This is policeman cat. Policeman cat is going to represent S-inositol, which is a mouthful. S-inositol is a drug that blocks NMDA receptors at the glutamate binding site. Do you remember earlier when I said that some data suggests synapse weakening can happen even when NMDA receptors are closed? Alzheimer's research is one of the primary reasons that we think that. When you add a drug that's going to block bad amyloid's ability to bind to NMDA receptors, like COPCAT, you're going to see synapse weakening, or you're going to see that the synapse weakening of bad amyloid is almost completely reversed, right? So here's bad amyloid by itself, and here's bad amyloid with an NMDA receptor blocker. When we put all of that together, we're going to find that bad amyloid blocks processes like the phosphorylation of CREB that are going to cause synapse strengthening, and it also causes synapse weakening through binding to NMDA receptors. These together are ultimately going to lead to the death of the cell, and neurons that aren't active enough will die off. Okay, that's going to bring us to the end of our discussion and my last topic for today. So, your brain dislikes too much or too little of anything, and that includes plasticity. It really likes to keep its functions within a homeostatic range. This is where the idea of homeostatic plasticity comes from. Side note, Dr. Torrigiano, one of the forefront scientists in this field, is my personal hero in science. That aside, if the cell is too active on average, all of its synapses will weaken by the same amount. If a cell is too inactive on average, all of the synapses will strengthen by the same amount. This is going to maintain the ratio of strength between two synapses. So if one is stronger than the other before, after homeostatic plasticity, it'll still be stronger than the other one. But it's going to allow the cell to be within optimal ranges for learning new things. So this is really how your brain stabilizes old memories and still stays plastic enough to learn new things. Okay, that is the end of this presentation. I really appreciate you guys letting me yap to you for a while. I'm happy to answer any lingering questions you might have, or if not, we can jump into some discussion. This is the cat you've heard yapping in the background. That's just in general. That just happens in your cells. You be quiet? No, she won't. So how does the inositol bind differently than the amyloid? So inositol is going to bind in a way that doesn't block that blocks the NMDA receptor without like changing its structure and opening it. So the amyloid is going to bind the same way that glutamate does, and the S-inositol is just going to bind in a way that doesn't activate the NMDA receptor. How are the hyperactive, overexcited neurons caused? So in Alzheimer's disease, so whenever neurons are, actually, we just talked about that. So when a neuron is too inactive because its synapses have all been weakened, it's going to kind of tip the scales and increase its activity. The problem isn't its activity, it's the fact that it's not receiving the signals enough. And so that means that it's going to kind of be overexcited, which is why we see that spike in activity, especially in the hippocampus, before we actually see symptom onset in Alzheimer's. Dopamine is a neuromodulator. So what dopamine does in those pathways is it acts kind of as a key. Okay, so your next question. So all neuromodulators will have a channel that is going to also act as like an ion channel. So nicotinic acetylcholine receptors. I forget the specific serotonin one. I think it's serotonin 5-HT3. Somebody might correct me on that. Dopamine is going to be D3. But in general, in the motor pathways specifically, dopamine is a neuromodulator, and that's because it's going to act kind of as a key. So in the normal pathway, you're going to have these excitatory connections, and the dopamine is going to come in and increase the excitation for what the brain wants to happen. And that's going to kind of smooth it out and reduce kind of the noise that your brain normally produces through just random firing. And then when we don't have that, we got all that noise, which is going to cause jerky movements or kind of like inefficient movements, which was also what we see with like cerebellar disorders and stuff like that. Like gaslight the interest? What? PTSD or thinking? So head injury and most traumatic brain injury is going to cause inflammation. That's the word, inflammation. And what inflammation does is it actually restricts plasticity. When cortisol binds to cells, it's going to have a a negative effect on the production of BDNF. Oh, experiential trauma. The only thing I could think of would be like stress, maybe. Stress, like a lot of stress would produce cortisol, and that would have a negative effect on like plasticity. We see that in depression as well. One of the like major theories for seeding of depression is that you have a lot of really high stress experiences, and those are going to decrease plasticity in the hippocampus by decreasing BDNF production. But that's kind of outside my field. Oh, no problem, Star. Just take your time. Stuff that promotes plasticity. So, really, any kind of learning, like we talked about earlier with Krebs, it's really just promoted by any kind of learning. Where is it? It's far back, geez. So Krebs is going to be activated by PKA, which is just involved in learning. It's also activated by calcium calmodulin dependent protein kinase, which is just happens when you have calcium influx into the cell during learning. And that Krebs is going to increase your adaptability for long periods of time. And it's going to also lead to the growth of new synapses. Pers and I were actually talking last night about Alzheimer's, and I made the point that adaptability is really important because if your neurons lose one connection from them being too weak, it's a lot more devastating if that's its only connection than if it like has 10 or even more. Skill regression, I don't think I touched on that. So dopamine is both. A neuromodulator is just a type of neurotransmitter that doesn't change the membrane potential. Oh yeah, thanks, Ryu. Let me know if you have any questions as you're taking that. So exercise is going to promote things like cell survival mechanisms. Um, Glucose processing is also really important. But the main thing is the cell survival mechanisms. There are certain pathways in your brain, or in all of your cells really, that are going to keep the cells alive. And whenever you do exercise, those pathways get activated so that your cells are kind of like more resilient, because when you are doing exercise, you are damaging your cells. It's very intensive for them. And so they kind of like bounce back. And over time, as you exercise more, that pathway is going to strengthen, and it's going to increase your overall resilience, including your brain. It's also just healthy in general. There's a lot of like connection between plasticity and your gut microbiome, blood-brain barrier, just diet, exercise, everything, because everything affects plasticity. Astrocytes wrap around synapses; those regulate plasticity with regulating the calcium environment. Microglia are going to eat away synapses, and if you don't have a hyperactive immune system, which is decreased sometimes by like exercise, then you're not going to have those like synaptic depression from the immune system activation. So a lot of things tie into plasticity. Neuroscience textbooks, we have um, a section for that. It's the resources section. I think it's pinned there. Wizzy might be able to say something about that. Is any type of exercise preferred over the other? So any kind of exercise will really do what you want. There are different kinds of exercise, like high intensity interval training, and they're all going to do different things. Woodsy can actually talk about this. We're both runners. There are different kinds of training, like zone 2 training is going to make it easier for your cells to like digest fat instead of glucose, which your brain really needs the glucose. And then like zone 5 training, which is based on your heart rate, is going to really increase the number of mitochondria that your cells have, which is really healthy for them for their energy production, and that kind of stuff. Is it possible to promote plasticity by any medication? It is, yes. We have medications called psychoplastogens. One of the big ones is ketamine. Another one is psilocybin. Those are the main ones that are being kind of researched in the field. There are also some labs that research, yes, ketamine is a psychoplastogen. It increases plasticity. That's why it's so good for PTSD and depression. There are also some medications that are being produced that have the effects. They increase plasticity, but they still don't. They don't have like the psychogenic effects. Like they're not going to make you trip out. Any other questions? Why isn't ketamine or psilocybin used for Alzheimer's? That's mostly because it wouldn't really do anything. Um, or well, it might do some things, but there's a lot more going on in Alzheimer's than just the plasticity. I just chose to focus on the plasticity because that's like my personal field. I could do a talk on Alzheimer's in the future because it's a huge field. Like we didn't even cover 1% of it probably. Um, but there are other things that are going wrong in Alzheimer's than just the plasticity. Are all psychoactive drugs plasticity increasing? No, there are a lot of them that don't increase plasticity. There's also a lot of them that actually kill your cells. I could do a talk on ASD in the future. Yes. How does acetylcholine make you focus? So it's a neuromodulator, which is going to do the same thing. It's going to be directed. Your brain sends it to specific places it wants to focus on. And then it's going to increase the excitability and make it easier for those specific neurons to fire relative to the neurons of the stuff that you don't want to focus on. So it's all about changing that signal to noise ratio. Oh, I haven't read that. I'll have to add it to my list. Thanks, Wizzy. Do you think ADHD medication helps in a more direct way to plasticity? That it facilitates learning? I honestly don't know. Plasticity isn't something that I've heard of as like deficient in ADHD. It's more the well, I guess it kind of is. I don't know. That's outside my field. I haven't done any research on ADHD. You do not have homework, no. Books to get started on. There are some. We have a resources channel. I sent it earlier. Um, I might add some books to it. I have a couple of favorites. Oh, here are my citations. Here they are that I promised. Hmm, that's interesting. It's really, really interesting because most of the time when we're talking about autism, we actually like we're talking about too much plasticity. That's what happens in the majority of the different like sub disorders within ASD. So it's really interesting that rigid thinking might be associated with too little plasticity. Am I neuroscientist? No, I'm Inka. How is hyperfocusing explained neurochemically? I do not know that. Sorry. Maybe somebody else can explain that. You explain things like an AD patient increasing plasticity and reducing beta amyloid can be a way to treat the disease. So it's part of treating the disease, vivid. Um, but like I said, and I guess I will do a talk on Alzheimer's in the future, it's only part of the disease. There's also like microglia hyperactivity. There's the like development of free radicals. There's neuroinflammation from the torsion that's put on the lobes as the tau tangles accumulate in the axons and cause them to bend. It's a really complicated disease, and I can really only talk about part of it when doing a presentation like this.
Yes.
Alrighty, well, I will stay a little longer just to chat and answer more questions, but that's all I have for today.